Oh, you guys are so sweet. Thank you. I think it's just because you got some light therapy and some grounding at lunch. Yes, all you barefoot people out there. So we are going to talk today about addiction. And you're probably wondering why an immunologist is talking to you about addiction. But this has been the big thing that has come out of the pandemic. We know that so many people are suffering from addictions right now. So that's what I thought I would focus on today. And I would bring my lens, which is looking at that psychoneuroimmunology component of addiction. But before we get started, we got to start with some disclosures. And as was mentioned, I need to disclose the first thing because Thena is a company that I'm working with right now, started by two naturopathic physicians in Portland, and it is a microbiome-based therapy. It is postbiotics. For those of you who don't know what postbiotics is, this is, their product in particular is a sterilized fecal transplant. So if you haven't talked to Thena yet, you should check it out because it's pretty exciting stuff. I also sit on scientific advisory boards for Burt's Bees and Clorox and GSK, and I am faculty at NUNM, University of Western States, and the Academy of Integrative Health and Medicine, and I speak for Metagenics, and of course I have my NIH funding. So I get money from a lot of different places. That's all that means. And we should all diversify, right? Yeah, I give you John and his book, Diversify. Yes, buy a copy. And Amy has a book too, so you got a lot of books coming out of this show today. All right, our goals today are comparing underlying mechanisms of addiction, discussing how the nervous system, the immune system, and the microbiome interact during addiction, recognizing some of the brain imaging patterns, and then discussing the link between emotion and addictive substance use. And that's how we're going to bring in that spirit component. So why addiction? As I mentioned, drug use is at an all-time high. Now, the drug popularity differs from nation to nation, which drugs are being misused, but if we look across the world, drug addiction, all-time high. We know that there are genetic, physiological, and environmental influences on not only substance use disorders, but also other types of addictions. So we're now seeing that with caffeine, sugar, gambling, sex, gaming, cell phone addiction, shopping addiction, that's right. There's so many right now, and it's really hard to figure out what all of the underlying things are that are contributing. Have we always had addictions? So I went back in history and I looked at 200 million years ago. There is evidence that we were using psychotropic plants. Wild, right? But we weren't addicted to them. And in fact, 40,000 years ago, we were using nicotine. And 13,000 years ago, people were chewing on betel nuts, but there was no addiction. So what happened? Well, first let's address this. Too often, the treatment of addiction simply works to alleviate the symptoms, dealing with overcoming the physiological dependence and working through withdrawal symptoms as the body readjusts to a non-dependent state of homeostasis. But that's not enough, is it? Because we know that just treating the symptoms doesn't work. We actually have to look at personality, behavior, and evolution. How did this person develop their addiction? So let's talk about evolution for a minute. It turns out that 200 million years ago, and 13,000 years ago, and 5,000 years ago, we had limited resources. We didn't have whole fields of poppies planted. We didn't have whole fields of marijuana or greenhouses filled with marijuana. And we relied on the limitations of the environment. What that means is that we never evolved to have a regulatory control over addictive substances. We don't have any internal control. We can't stop. we have not yet evolved to adapt to our modern environment of excess, shopping, leaving us vulnerable to addiction as we head into Black Friday season. And it's a season now, right? It's no longer Black Friday. All right, so here's some of the history which I thought was interesting. It turns out that ancient indigenous civilizations used psychotropic plants as food sources. It meant that they could forage longer, and it meant that there were tolerances to temperature changes and all sorts of environmental shifts that they could do if they were using psychotropic plants. They also got psychotropic plants as nutritional sources. So it was a common thing that was done. We co-evolved with some of the psychotropic plants, and a series of changes occurred between the mammalian brain and the psychotropic plants that allowed them to affect one another during the process of evolution. And in fact, Terence McKenna proposed the stoned ape hypothesis back in the 1960s, where he proposed that one of the things that was happening is as we moved from apes to humans, we were eating psychedelic mushrooms. And it's actually the psychedelic mushrooms that allowed our brains to evolve a higher level of consciousness. So what does that mean now when we have all these other psychedelic plants, right? Could we actually evolve away from the, I don't know what you want to call our modern society, but it's not good, right? All you have to do is watch one political ad and you know it's not good. I mean, I think we need to encourage a little bit more psychoactive substance use, but not addictive. So, okay, let's talk about some of the underlying mechanisms for addiction. First of all, with respect to chemicals, we have over 30 million chemicals, but only about 100 are addictive. Interesting, huh? So why are they addictive? Well, it doesn't matter whether we're talking about a stimulant or a sedative or an antinociceptive. All addictive substances are subjectively rewarding. They're reinforcing and they're pleasurable. And it turns out that they have the same effects on laboratory animals as they do on humans, in that order. So if something is more addictive in a human, it is more addictive in a lab animal as well. And if it's less addictive in a human, it is less addictive in a lab animal. So far, for everything we've tested with two exceptions, LSD and mescaline. Everything else has the exact same effect on the reward circuitry of the brain. So here is a brain. And what starts with an addiction is a trigger. Usually that trigger is a visual stimulus of something that the person is addicted to. So if you happen to be addicted to alcohol, it might be you're walking past a bar. If you're addicted to food, maybe it's the yellow arches of McDonald's, right? There's something that is a visual trigger. That trigger leads you to perception. And perception means you imagine yourself consuming that substance or buying that pair of shoes or winning that game. That imagining leads to the frontal cortex of the brain where you plan, how can I make this happen? And that planning then triggers the nucleus accumbens and the VTA in the midbrain to produce dopamine. And so just as you're doing the planning, you get the initial part of the reward. Then you crave. Okay, so that initial dopamine leads to a craving, and then that craving leads to you reaching for or walking to or getting on Amazon, and then you take action. So in every addictive process, whether we're talking about alcohol or cocaine or food or whatever, benzodiazepines, this is the process that happens. Now, I wrote it out in words because I didn't know if you went back to this, if you would go, oh, she said this, and then this, and then this. So this is just what I said in words. This pathway is stimulated overall by dopamine. So we call it the dopamine reward pathway. And the nucleus accumbens is the part of the brain that gives us that sense of reward. And it's the same sense of reward as like, you probably got your winter coat out because you didn't know it was going to be 70 in New Hampshire. And when you pulled out that winter coat, you reached in a pocket and you pulled out $20, right? And you're like, woo, $20. I forgot there was $20 in that pocket, right? That's the same dopamine reward system that you stimulate when you get high. So exact same pathway. Now here's the problem. It used to be that one cookie made you happy, right? And you'd go, woohoo, I got a cookie, yeah. And then you habituate. You develop tolerance over time. And as you develop tolerance, you need more and more cookies. And that's where addiction really starts to become a problem. So what's happening in the brain when you start to habituate? First of all, you're producing dopamine, and remember that dopamine is produced at a synaptic junction, and it's going to be released by one part of the nerve and accepted by a dopamine receptor. But when we have an addiction to something like cocaine, why is cocaine addictive and other chemicals aren't? Well, cocaine blocks the dopamine receptor that allows for down-regulating dopamine after it's been released. So in this figure, dopamine is in kind of a peach color and cocaine is in red. And what you see is that the cocaine is blocking the dopamine receptors on part of the synaptic junction. As a result, dopamine accumulates in the synaptic junction, and you fill more of the dopamine receptors, and you feel better and better until you downregulate those receptors. And once those receptors are downregulated, then you're like, oh, I don't feel so good. I think I need more cocaine, right? Meth has a slightly different activity. But it illustrates the other way that these addictive substances work. What meth does is it increases the amount of dopamine in the synaptic vesicles so that when you release dopamine, you release more of it. So you can either block the reuptake of dopamine, or you can produce more dopamine. And either way, you're going to get this big reward from having a drug. Different substances are actually activating different parts of the pathway. So some substances focus on the nucleoccumbens part of that pathway. Some substances focus on the ventral tegmental area of the pathway. Either way, it's that pathway of reinforcement of the reward pathway that's happening during addiction. Now, it's important that we have 5 different dopamine receptors. The reason that that's important is that it means that you can adjust to multiple different levels of dopamine. So if you only have a little bit of dopamine, you bind to dopamine receptor 1. If you have a little bit more, you bind to receptor 2. More yet, receptor 3, etc. So we adjust how we respond to our different neurotransmitters based on the receptors, the number of receptors and the receptor density. And that becomes important when we think about things like serotonin. There's 15 different receptors, which means you adjust to very small incremental changes in serotonin. Whereas with GABA, as an example, there's only 7 receptors. So you have more coarse reactions, and the same is true for dopamine. What we see in addiction is what we're able to stain in the brain are the dopamine receptors. So if you look at a normal brain, you're going to see the dopamine receptors, and you'll see them on the left side, yellow and red. And as somebody becomes an addict, they downregulate dopamine receptors because they're making so much dopamine. And every time dopamine binds to dopamine receptor, it's going to downregulate. So the brain of a meth abuser has much less dopamine receptor. Now you would think, okay, so they're less responsive to dopamine, so they're going to want less meth. No, it means they want more meth because they're trying to flood whatever dopamine receptors they do have. All right, let's bring in the rest of those systems. So it's not just dopamine. It's primarily dopamine involved in that reward circuitry, but it turns out serotonin is involved, the opioid receptors are involved, the endocannabinoid system is involved, GABA is involved, and glutamate is involved. And so all of these different neurotransmitters are getting involved when we talk about addiction. And so do cytokines. So interferon gamma, IL-6, and TNF-alpha have all been implicated in addiction. We're going to talk about each of these in a little bit more detail here in a second. And then your microbes of your gut play a role. Short-chain fatty acids have been shown to play a role, and microbe-derived neurotransmitters play a role. So again, just a reminder that your microbes make a bunch of your neurotransmitters. They're not all made in the brain, and we'll go through that in a little bit more detail. And it shouldn't be a huge surprise to us that the microbiota is involved. It seems to be involved in everything, right? It's like we didn't even know about the microbiota 10 years ago, and now it's responsible for everything, right? I mean, that's how I feel about it. I'm like, what, 10 years ago I knew E. coli, and now I'm supposed to know all these different microbes? Okay, come on. Like, it can't be that different, but it turns out it's probably pretty relevant. So what happens is that the microbes in the gut produce neurotransmitters. Those neurotransmitters travel around the body, and I believe it was Dr. Tom this morning who was talking about the fact that serotonin is carried from the gut on platelets to the brain, and then you actually have transporters to transport the serotonin across the blood-brain barrier into the brain. So just because it's made in the gut doesn't mean that it doesn't have an effect on the brain, and of course it does. Same is true for dopamine. You make dopamine in the gut, and it's going to be transported to the brain. So for sure, whether or not you have dysbiosis in your gut is going to influence how you might react to an addictive substance. If you have dysbiosis, you're actually, well, we'll show you. I don't want to give away the punchline. All right, so behavioral symptoms of cytokines. Remember this morning when they were talking about how come, and Dr. Tom was talking about this too. I learned so much from Dr. Tom, right? Yeah, so how come when you get sick you want to go to bed? I was back in the corner going, IL-1, that's why. You want to go to bed because of IL-1. Because the first cytokine you make during an infection is interleukin-1, called interleukin-1 because it was discovered first, because it causes fever. It goes to the hypothalamus and it causes fever. But IL-1 also makes you sleepy. It makes you malaised. It makes you tired. You're supposed to keep your ass in bed, right? But do you? No. You get up and you go to work because IL-6 makes you anxious, and you're like, I can't miss work today. I have too many things going on. Oh my God. Like, I can't miss work. And so our anxiety is like, no, we've got to keep doing things. Those relationships are bidirectional, which means that if you are feeling depressed because you lost someone you love, because, I don't know, they didn't have the pair of shoes you want, whatever it is, you will produce IL-1. Likewise, if you're feeling anxious, maybe you're anxious because you're stuck in traffic on the 95 or something, I don't know. Fortunately, I don't live out here anymore. Don't get me wrong, I love this area. How can you not love it? I mean, look out the window. But I did not like commuting on the 95, that is for sure. Anyway, because you're like, oh my God, I got to get to work, right? I'm anxious. IL-6, through the roof, right? So it's bidirectional. If you experience anxiety, you will produce IL-6. If you produce IL-6, you will experience anxiety. That's a positive feedback loop. That's not good. How do you break a positive feedback loop? You have to intervene somewhere. And so that's where doing some mind-body medicine, doing some herbal medicine, doing some grounding might help reduce that inflammation. And TNF-alpha is interesting as well because TNF-alpha is what makes us hostile and aggressive. This has been studied in stockbrokers. I know, doesn't surprise you, does it? It's also been studied in hostile marriages. It's been studied in athletes who are aggressive. TNF-alpha correlates with aggressiveness. Now, I want you to think about some of the addicts you might know. Do they get aggressive? Yeah. It's inflammation. That's what they're experiencing. And I think we often will go, oh gosh, I'm afraid of that person because they've become violent. What they've become is inflamed. And we have to remember that, because as healers, if we can hold the space that this is inflammation and we know how to treat inflammation, then we can actually help them through that process. Why is this happening? Well, it's happening in part because of the kynurenine pathway. If you have increased inflammation, you have an increase in IL-6, and both IL-6, IL-1, and TNF-alpha will shuttle tryptophan away from serotonin and through the kynurenine pathway such that you make neurotoxic quinolones, right? And so by converting tryptophan to quinolones instead of serotonin, there goes your happiness. And remember, serotonin, ultimately, the secondary metabolite is melatonin, so therefore you can't sleep. So we end up with this inflammatory picture, which explains a lot of the symptoms people feel. Now, it's not just the pro-inflammatory cytokines. It turns out other cytokines also have an influence on our mood states. So interferon gamma helps provide our social behavior. And TGF-beta is an antidepressant. So, and that's good, right? TGF-beta is the primary cytokine made in our gut in response to food, and it's supposed to make us happy. And you just... Thank God it's Friday. Because it's thank God it's Friday. There you go. I love that. I'm going to use that. Thanks. I just thought you were happy with my dog picture. So this is a study that was done in Norway where they took 80 people with substance use disorder, and they looked at their cytokine profiles. And what you'll see is that if people were experiencing distress, their IL-6 was high. Not a surprise, right? We just learned why. If they were experiencing anxiety, their interferon gamma was low. If they were experiencing depression, their TNF-alpha was high. Anxiety was also associated with low IL-10, and then depression and distress with high IL-1, IL-6, TNF-alpha. I mean, it's the exact same thing that we've just talked about, right? So we know inflammation is involved in substance use disorders. Now, the neurotransmitters are involved as well, and we've talked a bunch about dopamine, which is that reward neurotransmitter. But we also have to remember that dopamine is also involved in pooping, right? So contraction of the colon, that's dopamine. That's why you make it in your gut. Serotonin is our happiness neurotransmitter, but serotonin is also involved in bowel movements, which is why you make 95% of it in your gut. GABA, our relaxation neurotransmitter, which we make the most of in the middle of the night, is involved in numbing the pain of pooping, because pushing poop through that little tube, turns out, can be pretty painful. I always laugh, is it a Colace commercial, if it feels like you're pushing through a pineapple? Yeah, if it feels like you're pushing through a pineapple, yes, you are probably dehydrated, but you also probably have low GABA, right? So good way to diagnose GABA deficiency. If people hurt when they poop, they are probably not making enough GABA. And then acetylcholine doesn't have a pooping effect, but acetylcholine has an effect on our calm and our anxiety regulation. So if we put it all together with our neurotransmitters and our cytokines, dopamine, we think extroversion, reward, and we're thinking higher levels of interferon gamma. Happiness, we're thinking serotonin, which means lower levels of IL-1 and IL-6, because if you have high IL-1 and IL-6, it's going to shuttle away from serotonin in the kynurenine pathway. Relaxation is GABA. That's going to be higher TGF-beta. And calm, acetylcholine, we don't know what cytokine yet is associated with that. So this gives us this lovely little pyramid where we see the nervous system and the endocrine system and the immune system are all interacting with each other. And when we have a stressor, we throw all of those systems out of balance, not one. And that's why I love, well, it's one of the reasons I love naturopathic medicine, is that you can come at this stressor then from any different direction. A variety of therapies will work because you could have an effect on the nervous system and that'll have an effect on the immune system. Or you can have an effect on the immune system and that'll have an effect on the endocrine system. But let's bring in the microbes. Let's bring in this new system that we haven't been talking about all that much. I love this. This is a picture of actual microbes in the microbiome, electron scanning microscopy, and then you can see the little poop there too. Because I think that we don't, right? I mean, we draw all these pictures and we don't actually know what it looks like. Actually, we do know what it looks like. It's pretty cool. All right, so I used a couple of new terms, so I want to explain them. When we ferment non-digestible fibers, we produce short-chain fatty acids. That's not new to any of us. What might be new is some of the terminology. So prebiotics is the fiber. We know that, right? But it turns out prebiotics is way more than fiber these days. We now know that polyphenols serve as a prebiotic, so every spice is a prebiotic, which is every medicinal herb, essentially, right? And we know mushrooms, the beta sheets in a mushroom act as a prebiotic. Yeast is a prebiotic. So lots of different things are starting to qualify as prebiotics based on their ability to make different sets of microbes grow. Probiotics are the microbes that are able to ferment the prebiotics. So probiotics we think of as the live microorganisms. We think of them as in a bottle, right? But what probiotics then produce is a postbiotic. And it turns out that those postbiotics are pretty dang important. Short-chain fatty acids are one of our postbiotics, but we have many, many, many postbiotics. So if we just look at human stool, there's well over 800 different postbiotics. Let's look at them. Acetate propionate butyrate, there's our short-chain fatty acids, right? But then there's the branched-chain fatty acids and the kynurenines and the indols and the indol derivatives and the tryptophan derivatives. And look, there's serotonin and dopamine and histamine. So neurotransmitters are actually postbiotics. It turns out that there's a lot of things that we used to think that human cells make, and our microbes make them instead. Now, why is that important? That's important because we give people antibiotics like candy in this country. And when we kill off microbes, we are killing off their ability to produce these important metabolites. They can't make neurotransmitters. They feel crappy, right? When we eat preservatives, we're killing off microbes. Yeah, the preservatives are designed to kill the microbes in your food, but if they kill the microbes in the food, what do you think they do to the microbes in your gut? They kill them. Yeah. And we now recognize that all of the chemicals we're exposed to, there's a great paper, a Nature paper came out in 2018 out of Germany that showed that non-microbial medicines actually act as antimicrobials. Our SSRIs are killing off the microbes in our gut. Uh-oh. Now what? And one of the reasons we know metformin works is it actually kills some microbes and makes other microbes grow. So it turns out that a lot of the medications that we designed because we thought they were going to have an effect on human cells are actually having an effect on the microbes in our gut. And it matters then what our microbes are making. So I've given you a list here of which microbes make which neurotransmitters. I'm just going to point out the top row. Lactobacillus and Bifidobacterium are two of the most common microbes we find in probiotics. Right? You agree with that? Yeah. So is it any wonder that people feel better when they take a probiotic? Because even if the probiotic is not alive, and we know that most probiotics are not alive, in the bottle, The lactobacillus and the bifidobacteria made GABA. So when you're taking that probiotic, you're taking GABA. Well, I can relax now. Okay, so what's our relationship back to addiction? Researchers took mice that had been treated with antibiotics, and they had a different gut profile because of their antibiotics. And they showed that if you don't have that particular microbe in your gut, you have an increased preference for cocaine. And you're also more sensitive to cocaine. Now, if you give those animals short-chain fatty acids, it decreases their preference for cocaine. So the metabolites are determining the preference for the drug. We didn't see that one coming, but it turns out it's probably true with humans as well. So here's our relationship. Dysbiosis, pre-substance use, then you start using a substance and you might get additional dysbiosis. That's going to give you this compromised gut barrier, which is going to change your gut-brain interaction and give you dysregulated brain-derived neurotropic factor, which in turn dysregulates your dopamine in your brain. And that addiction pathway completely affected by dysbiosis in the gut. And I think I just threw this in here so you could see what does it look like, gut-brain reaction. So here's where we're at. We got dopamine, neurotransmitters, cytokines, and microbial metabolites all having an effect on addiction. Now, what's the first thing people did during lockdown? Do you remember what we ran out of in the grocery stores? Okay, after toilet paper. Flour, we ran out of flour, right? They baked. You guys are smart, though. Toilet paper was right on the tip of your tongue. Right, so we ran out of flour, so everybody starts baking. They're all eating wheat, right? What's our problem with wheat in this country? The way that we process it, the pesticides that are on it, put on it after we harvest it. So there's more pesticide on wheat than on other foods. We also know that they're overusing hand sanitizer. That's going to cause dysbiosis right on the skin. Like, do we have dysbiosis from the pandemic? Dang sure right we do. And now add to that the stress and the anxiety, and we've got perfect storm for substance use disorders. Okay, so I'm just going to do a couple more brain images. I showed you that dopamine receptors are downregulated during abuse, so this is a different example. Normal person on the left, cocaine user on the right. But I think what's interesting here is that abstinence, if you can get somebody off of a substance, those receptors come back, right? They're recycling. So they can come back. So if you compare to healthy control, after 14 months of abstinence, someone is pretty darn close, which suggests there's hope there. So in the 3-stage model of addiction, you start with binging, and then you move into the negative reinforcement, and then anticipation and preoccupation. And it turns out that we can see each of those stages just by looking at the brain. So we see the binging at the top, we see the withdrawal and the negative affect in the middle, and then the preoccupation at the bottom. And what I think is interesting is that they're each happening in different patterns in the brain. So it's actually very identifiable. So here's where we are. MRI can demonstrate substance use in all stages, and abstinence allows us to repair those pathways. Okay, I saw this TED Talk once, and it was really interesting because it talked about the fact that we can put your grandmother on an opioid after she has hip surgery, and she's not going to become an addict, right? There's something else that's going on. There's an emotional component that's going on. And Portland, Oregon is in a very interesting place right now because we decriminalized all drugs. Probably was a mistake. Here's why. we were following what they did in Portugal. And in Portugal, they decriminalized all drugs. And at the same time, they put in programs for people who were drug abusers to get grants to start businesses, to get scholarships to go to school, to have different things to do with their lives, to develop life purpose, right? And once they have life purpose, they don't go back to using the drugs. So if you have good things going on in your life, you can take an addictive drug and not become addicted. But if you don't have good things going on in your life, then you take an addictive drug and then you become addicted, and many of them are now living on the streets in Portland. So it leaves you to this question of why are some people more prone to addiction? Like, where is that coming from? And this goes back to what, again, Dr. Tom was talking about. It's coming at a really young age. Now, we know that there are some genes that predict addiction, and the current guesstimate is that 50% of addiction is genetic and 50% is environmental. We do genetic-wide screening association studies, and we also look at single nucleotide polymorphisms. And so I'll give you the example of cannabis addiction. If they do genetic-wide screening, they find that there is a spot on chromosome 8 that is associated with a particular protein, and if you have low levels of that protein, you are more prone to abuse cannabis than people who don't have that protein. With alcohol, there's been 14 different genetic loci involved in addictive behaviors. I didn't start I brought my clock up so I could start it, and then I didn't start it. How many minutes have I got left, Cora? Great, awesome. Okay, so we know that some of the genes that are involved in alcohol addiction are involved in metabolism of alcohol. So you see like the aldehyde genes and that sort of thing. And then some of the genes involved in alcohol addiction, they see are more widespread. So they're involved in tobacco use, caffeine use, cannabis use as well. We're now starting to look at those genes for things like eating disorders, gaming disorders, et cetera. So why are some people more prone? Well, there's a genetic predisposition, but there's something going on with personality and emotion as well. And that we know that emotion can predict the intensity of emotional state, and it's a well-established influence on substance use. So people who are not good at regulating their emotions are more likely to engage in substance use or to distract themselves from emotional states. So here's what it looks like. You experience an unpleasant emotion, and I'm sure many of us have felt like this. You use a substance, and it distracts you from that unpleasant feeling, right? You get immediate, short-term gratification. The downregulation, I pulled this from a paper because I thought this was an important statement. The downregulations of emotions is an effortful and not necessarily immediately rewarding process. As the need for regulation persists, individuals experience a shift in motivation from the regulation of emotion toward the acquisition of rewarding experiences. In turn, there's an increased allocation of attention toward cues that signal immediate reward, thereby increasing substance use. So people have started studying this, and what we've seen is that individuals with alcohol and drug use disorders have been shown to have greater deficits in their emotion regulation. There's been cross-sectional and correlational studies that have found greater deficits in emotion regulation relate to worse substance use. The harder it is for you to regulate your emotions, the more likely you are to find a substance that will help you. And clinical research is providing evidence for the impacts of emotion regulation on substance use and treatment outcomes. This is a study that was done where they compared adults with clinical addiction, so they'd already been identified with addiction, and healthy college students. And if you look down the right-hand column, just look at the p-values, and then look at the left-hand column, you'll see that a negative overall affect is very strongly associated with substance use. Negative goals, negative impulse, negative strategies, all of those things very strongly associated, p-values of 0.0001 and lower. Whereas adaptive strategies, not associated at all. If you could adapt, no substance use issues. What about maladaptive strategies? Highly associated. So we see this correlation happen. Now, that was done with people who were in their 40s and 50s. We're now starting to look younger, because one of the things that we saw during the pandemic is we saw that 18 to 26-year-olds were the most depressed from the pandemic. These were the folks who were graduating from high school, graduating from college, supposed to go out and do jobs, and there were no jobs for them to go to. So identifying that they were highly depressed, are they more likely to become addicted to a substance? And what we saw is that the emotional regulation is highly correlated to their attachment. So now we go into attachment theory. And non-substance related addictions like gambling, video game addiction, and problematic internet use was also much higher in people who did not exhibit good emotional control. So we see emotional regulation is a predictor for all addictive behaviors, both substance and non-substance behaviors. Emotional control is the most powerful predictive for addictive behavior. Poor attachment predicts gambling disorder, problematic internet use, and video game addiction. And females score higher in maternal attachment and peer attachment, whereas males score higher in relation to gambling disorder and video game addiction. I don't know if that's a surprise to me. I got married in May. I know, I'm a late bloomer. Took me till I was 53 to find somebody, but finally found him. But we've got 5 kids, four boys and a girl. He has 5 kids, I have none, but now I do. But with 4 males between the ages of 22 and 29, this does not surprise me. So, does secure attachment predict addiction? It turns out that folks who have secure attachment are characterized by self-acceptance of their own emotional needs, whereas individuals with non-secure attachment tend to be more anxious and avoidant behaviors. They pay little attention to their emotional needs, and they do not feel that they can rely on somebody's support. So attachment, when we are infants, strong predictor of whether or not you're going to develop an addiction as an adult. This becomes really interesting, and this figure's a little bit busy, but I want you to start by looking at the left side. If we look at the left side, look at the fact that the sensory input is an infant looking for face cues, okay? So you've got your infant looking to their parents for facial cues. That's triggering oxytocin. It's not just breastfeeding triggering oxytocin. That's triggering oxytocin, and that is serving as the stimulus of the dopamine reward pathway for an infant. So you staring at that baby and making eye contact and facial cues, and let's admit it, we all do it. Oh, you're so cute, right? That's stimulating that dopamine reward pathway in that infant. Tell me what's happening today with making eye contact with our infants. Cell phones, masks, We're not giving the same visual cues to infants today. And we haven't been giving the same visual cues. In fact, there was a really interesting study showing that all of these gizmos we have that put the baby facing backwards, right, on us, instead of facing our face, the baby's on our back or the baby's on our front, but it's facing out, we're not allowing the baby to have the visual cues from mama that it's used to. And we're having less secure attachment patterns as a result. So maternal turns out to be more important than paternal. And child neglect doesn't necessarily mean that you left your kid in the back seat of the car and you went to the grocery store. It can mean that you're playing on your phone and your child is over there not having visual facial cues. The infant processes those cues. You see a reduced activation in the dopamine pathway, and as an adult, they develop substance use. Doesn't even have to be as an adult. We're now seeing that children are getting that dopamine reward from videos on television. You know, why are they watching The Jungle Book 35 times? Because they're getting that dopamine reward pathway stimulated by the television instead of by their parent. And that's one of the pieces that's now feeding into our addictive behaviors, and the fact that they could have a gaming disorder by the time they're 7 years old, right? So lots of things for us to be thinking about as we're thinking about prevention of all of the things that people get as an adult. And again, why I just loved Dr. Tom's talk, because it does go back to everything that happened when you were a child. So how are we going to fix maternal neglect? Okay, this is a study done by conventional biomedicine, of course, right? We're going to give mama intranasal oxytocin and hope she bonds with her baby. We're going to give the baby intranasal oxytocin, and maybe then it won't need maternal cues. Or maybe we address mom's own attachment history and explain to her how important those visual cues are for downstream growth of their child. Now, people don't take addictive drugs forever. It turns out the average time that someone is on cocaine is about 4 years. The average time someone is on marijuana is 6 years, alcohol is 15, and nicotine is the hardest to get off of, and so that's 25 years. The projection is that marijuana, the number of years are going to go up now that it's much more accessible, that this research was done when marijuana wasn't as accessible as it is now. So to summarize, dopamine is mediating the reward experience from addictive behaviors, but cytokines, neurotransmitters, and microbial metabolites are all contributing to addiction. Brain scans of addiction highlight the absence of dopamine receptors. Addiction is about 50% environmental and 50% genetic, but our ability to regulate emotion is very strongly related to addiction. And maternal attachment is related to the emotional regulation and those healthy dopamine reward pathways. There you go. Thanks. Do we have any questions? Yeah.
I thought that was amazing.
Thanks.
That was great. Well put together. Said a lot about science and integration of gut and brain health. Loved it. Question. You said dopamine binds to receptors and that receptor is instantly downregulated. Are you implying that a single use of a neurotransmitter binding to a receptor removes that receptor from the equation and that you can...
Temporarily, yes. So usually what happens is dopamine binds to the receptor. It triggers endocytosis, and so that particular receptor will go into a recycling pathway, and then it'll show up back on the surface, usually somewhere between 24 and 48 hours later. So even single use, you're going to get a temporary downregulation of dopamine receptors. Yep. Interesting, huh? What's really interesting to me about that is that that's true for most receptors, right? So we were doing a study where we were looking at alpha-lipoic acid for MS, and we actually found that the best administration, this was in mice, it was a mouse model of MS, but we found that the best administration was not daily. It was every 2 days, because you give the alpha-lipoic acid and then the receptors go into a recycling pathway and they're not available the next day. And you actually then have receptors again 48 hours later, and you get your best effect if you do every other day. So I asked one of the docs that I was working with, well then shouldn't we be prescribing herbs and whatnot every other day instead of every day? And the issue is that if you don't tell people to take them every day, they don't take them every day. They forget. And so if you tell them every day, then they'll take them and they'll remember to take them every day. They get into a routine, but you actually probably get a better effect from every other day or even every third day.
With most things?
With anything that has a protein receptor, yeah. Interesting, huh? Yeah, I know, science. What are you gonna do? Anything else? Over here.
Just because, mothers are at fault for everything that happens wrong
Oh,
with their
I know.
children. I just
know.
was curious about where the difference between maternal and paternal input was. Just that moms were around more, or that mom input was paired with breast milk or something like that?
Yeah, so they can't figure out why it's more maternal than paternal. And the same group did like 3 follow-up studies to try to figure out, like, is it just that mom is around more? But they actually looked at scenarios where mom died in childbirth and dad was a sole caregiver, and the baby still did not fixate on dad as much. In the discussion section, they talked about whether or not males have the same facial expressions as females, and was there a difference in facial expression between moms and dads, but they didn't actually test it. So I don't know. They're still in a theory phase there.
Did they look at pheromones?
I mean, your guess is as good as mine at that point, right? Yeah, it is fascinating. I'm working on my own book. Seems like everybody's writing right now. So anyway, one of the docs that I'm working with, he was in medical school when they gave birth to his son, and his wife was in nursing school. And his son has gaming disorder. And he said, we have wondered from the get-go what happened. And my wife was never around because she was in nursing school and I was in medical school, so my face was in a book the whole time. And our child was essentially in his carrier under my desk, and I would rock him with my foot. And he's like, yeah, I can believe that there was no facial cues whatsoever. So it was interesting because it kind of illustrated what this study shows. But I wish I had a better explanation. I don't. Yeah. There's a question over there.
Sorry, who's it? Oh, is that you? Sorry, who had the question? Oh, okay.
So in terms of the mother, it's a twofold question. Mm-hmm. So in terms of the infant and the mother facial cues, what period of time does that include up to? So say somebody's in an orphanage for the first 7 months of their life. Is that like they missed the boat?
Yes. So that particular study in that group is studying the first 1 year of life. And the first 6 months are critical.
Okay. So secondly, that then goes back to addressing the mother's own attachment history. So what are some of the considerations that are part and parcel of that topic?
So they're doing family-based therapy for that study, where they go. I have never done family-based therapy, so this is just based off of my reading. But what they've been doing is they have a group of individuals who are all doing this family-based therapy, and certain individuals stand in for a parent, a sibling, etc. And then the person is discussing like what they recall about those relationships that they had with their family and their parents and whatnot as they were growing up, and what their attachment was or was not with their family. Again, I don't know if it's going to be effective yet. It's what's reported in the literature, and everything I'm telling you about is like last 5 years. So we'll see now post-COVID as some of these studies get completed and reported.
And then lastly, one of the things that's becoming a pretty big topic is adverse childhood experiences as it relates to all of this,
Yep.
addiction behavior as well. And I wondered about your kind of not going there in this particular presentation, but if that's something that you've thought about for your book.
It actually is, because our book is on stress and immunity, so yeah. And I actually have a postdoc who works on adverse childhood experiences and inflammation later in life. We know that ACEs strongly contribute to a state of chronic inflammation. You see higher levels of IL-1 and IL-6 in particular in most individuals, and higher TNF-alpha, not as much in everyone, but in some individuals. And so we're seeing, so the question is why, right? Why do those genes stay perpetually turned on? Because you should turn them off eventually. But this leads to an epigenetics question, right? So if you're regulating the gene, if you're turning it on and off, that's epigenetics. And I was just part of Jeff Bland's think tank for microbiome stuff. Michael Skinner's research is really interesting. He's at Washington State University, and he's been looking at gene regulation and epigenetics, and he thinks that you combine the adverse childhood events with environmental toxicity. So now add BPAs, add diesel fumes, add, name your favorite, he looks also at glyphosate, but name your favorite environmental toxin. And is that changing such that those genes just never get turned off? You also have Julian Thayer, and he looks more at racism and ACEs combined with racism, and looking at continuous exposure to microaggressions. And he can show that, again, cardiovascular genes in particular are showing upregulation in the vast majority of folks who have experienced racism after experiencing ACEs. And interestingly, his stuff is, he shows, which I think we probably could guess, but he shows that some people become hypo-responsive to stress, like you need a larger stimulus to stress them out if they experienced ACEs, whereas other people become hyper-responsive to stress, meaning that they experienced ACEs and now they're, hair trigger, and you're going to turn on the inflammatory pathways. And all of that is under epigenetic control. What Michael Skinner's data is showing is that your great-grandparents influence how those genes are how those genes are regulated for you as an adult. Which means, are we ever really going to know? I don't know. Super interesting stuff, though. But yes, ACEs are absolutely involved.
We'll take just one more question.
Thank you, Heather, for the. Oh, you're going. I'm going.
I heard someone.
Go ahead.
I thought Alyssa was going. I have a question. So do you know that if antibodies made against neurotransmitter receptors also have that same downregulating effect over time? Like especially dopamine or glutamate?
Antibodies to neurotransmitter receptors?
Yeah. The antibodies binding instead of the actual neurotransmitter?
I don't know. It's a good question. Yeah, I don't.
Thank you.
I don't know.
Okay, I guess since that was a short answer, they can scoop
Then we
me
can
in
do
for
one more?
We do one.
Phenomenal presentation. Thank you as always for lecturing. In patients with ADHD, where the dopaminergic system is already affected,
Yep.
they're at higher risk already for addictive behaviors, unmedicated ADHD patients. Can you talk a little bit, are you familiar with, in those patients then who become addicted and then they stop, that in the slides that you showed, in the MRIs, the dopamine activity, there was a signature still there, and the neurons would, as I understood it, repopulate. What about with somebody who's already neurodivergent to begin with?
So that is a really good question, but not from the receptor perspective. One of the things that is coming up with ADHD, autism, some of these other things, is that one of the major defects is in the gut, right? And if you don't have the gut microbes producing the neurotransmitters, then you're going to wind up with dysregulated nervous system overall. So with ADHD, if you were to repair the gut aspect so that you can actually produce the correct amounts of dopamine and serotonin and glutamate, which is one of the big ones that's misregulated in ADHD, you actually can get the child to stop exhibiting that behavior. We've been doing a really interesting study with Jenny Johnstone, who's at OHSU. She's been using it's a 40 micronutrient supplement. It comes out of Australia, and they used to use it for tail-biting pigs. I know. And somebody said, hey, this works on aggressive pigs. I wonder if it would work with aggressive ADHD kids. And so they've done 3 clinical trials now, and it works, right? But it's repairing which microbes. And what's really interesting is because we're doing the microbiome analysis right now of these kids, and what matters is the starting place and the ending place for the kid, whether or not they got the micronutrient or not. So the placebo kids, some of them got better, but it turned out that the placebo had rice flour in it. And of course, rice flour is prebiotic. And so it's having an effect on the microbiome. And we're seeing that for kids who got the placebo, their microbiome moves this way in a PCA analysis, whereas kids who got the micronutrient, their microbiome moves this way in a PCA analysis. But either movement is therapeutic. Either way, it works. So it's interesting because I don't think, again, that we thought about the fact that the microbiome was producing neurotransmitters, and if we fix the gut, we would actually fix the ADHD behavior. Yeah, they do. It's not just the microbiome. You're right. There's a whole lot of neurotransmitters in the gut. Yep. Okay, I've overtalked. Bye, y'all.